Thank you very much, Steve, Barry, for inviting me back. I will share my screen now. Here we go. Yes, whatever did happen to influenza? I don't know about you, but occasionally I give my opinion quite freely and loudly to the television newscaster or the expert that turns up when they say something that I perhaps don't entirely agree with. And I've shouted at the television a couple of times over the last few years, usually in mid-August, end of August, when somebody pops up and says it's going to be a terrible, even worse autumn because flu's on the way and we're going to be really inundated and have a dreadful season. And I said to myself and to the newscaster that I didn't think that was entirely accurate. And as it happens, I was right. But what did happen to influenza? Let's have a look and see. First of all, we know the anecdote and hasn't been around. What's the evidence? Most countries around the world, and certainly developed countries, have quite sophisticated surveillance systems, taking throat swabs and nose swabs from people with influenza-like illness, testing them by PCR, a sensitive way of testing for the virus particle itself, and then reporting that to either a national surveillance system and also to international surveillance system. And this chart is from the WHO. They run a global surveillance system called FluNet. And if you look on the left-hand side, the blue columns are influenza positive samples that were reported to WHO. Need to get rid of that. And then this is the winter of 2019-2020. And flu, very helpfully, of course, usually spans two calendar years, December to January, February. And 2019-2020 was a very ordinary year for flu around the world. And the evidence, the reports demonstrate that. We then come to 2020, 2021, and indeed the beginning of 2022. And the blue line is almost baseline, completely flat. Very, very few reports of positive samples of influenza. And as you've seen from the note at the top, the red line is COVID. And COVID-19. So completely taken over, but influenza was not reported. My screen won't move on. That. This is another chart from the World Health Organization, which goes back a few more years, from 2011. Every year we get some sort of influenza outbreak, and these are the reports that were sent from England to the World Health Organization in their FluNet surveillance system. And then we have 2020, beginning of the year, very high levels, and then nothing reported. If you look at the top right-hand graph here, it is the reports for the UK, probably England, that is for the last 4 years. The grey line here is the isolation of identification of flu, a particular strain, the strain that tends to cause the most severe illness. And it was quite normal for 2018 to 19. 2019 to 20 was an even higher level of influenza infections. Influenza has a very precise definition medically. It's a sudden onset of symptoms with at least one of fever, malaise, feeling unwell, headache, or chest pain, plus at least one of a cough or sore throat or shortness of breath. So that's an internationally recognized definition of what we mean by influenza-like illness. The surveillance figure that I like most is this one based on GP consultation. So these aren't positive laboratory confirmation of flu, but people coming into the GP, you remember that scenario, this huge line here is 2010, 2011, and we'll come back to that. And these are the rate per 100,000, a big spike. 2019-20 was nothing like as big, but it was substantial. Again, 2021, 21-22, the numbers of consultations for flu, even given the difficulty of getting in to see your GP by whatever means, is very low indeed. So all the evidence confirms that flu did practically disappear from circulation. And the UK or the English surveillance system, even up to a couple of weeks ago, which is the most up-to-date one, the number of flu isolates confirmed in the laboratory is very, very low at the moment in England. So demonstrably, flu did decrease significantly over the last couple of years. So why? That's not rocket science to work this out. We had no international travel over the last 2 years, 3 years almost. We had very little social mixing in the winter time. Those were the times of the lockdowns and asking us not to socialize. We were wearing masks. It's a respiratory illness. So our lack of social mixing really did make a big difference. Also, we were very strongly encouraged to have our vaccine, and I'll be coming back to that obviously later on. So there was a high level of immunity to recent strains in the community, and all of that added up to very few new strains coming through, and the old strains weren't circulating. So as far as the humans were concerned, flu has not been a widespread infection worldwide over the last 2 winters. But there has been a huge outbreak in the avian population, one of the worst that has been seen around the world. And it's called avian influenza. And there are 2 different sorts of avian influenza. There's the highly pathogenic avian influenza, and we'll come back to this notation in a little while. And there's also a lower pathogenic avian influenza. They both infect all sorts of birds, excuse me, Wild birds, wildfowl, migratory birds carry the virus. Some of them have higher levels of immunity and are not usually as sick as domesticated agricultural animals that we keep. Domesticated birds, in fact. But the wild birds can pass the virus on to domesticated flocks. And it is well known that the migratory birds spread avian influenza virus all around the world. This chicken looks as though he's sick. They get droopy wings, they get sneezing, secretions, but avian influenza is not primarily a respiratory virus. The virus replicates in the gut and is present in the fecal material from birds. And as you know, avian feces are very dry and they particularize quite easily so that they spread through the particles and are taken in by other birds. It is has been a very significant time for the UK and indeed through Europe. And we have had what is called an avian influenza protection zone, an AIPZ, put in place in the UK for about 18 months, which meant that all domesticated birds, including the commercial ones, chickens, ducks, turkeys, and also pet animals, have had to stay indoors. So that you've probably noticed in the supermarkets, they've been able to relabel the eggs, not as free range, but as perch laying, because the chickens were not allowed out into the countryside. All this is to minimize the contact with the wild birds that you can't keep under control. Because if a flock of birds is identified as having influenza, the whole flock is culled. It's got huge implications commercially. And also environmentally. This is a picture taken on the Farne Islands, where they have found through surveillance that the virus, this highly pathogenic strain which had made its way to the UK and through Europe, was killing some of the birds. And the Animal and Plant Health Agency surveillance says that if you should send in birds if you find 5 or more dead birds of any species together, and wildfowl, so that they can monitor what's going on in the wider natural environment. And the Farne Islands had a severe outbreak. According to the figures on the APHA website, over 1,400 wild birds were found to test positive. So it's widespread, even though some of the wildfowl don't get sick. It is widespread and does kill wild birds. That was over 347 different locations in the UK, and it covered 61 different species of wild birds. So it's been a very significant outbreak. Again, there is European surveillance of the highly pathogenic avian influenza virus. The peak you can see here on the left-hand side is 2016-17. Again, a lot in the winter time. It's when a lot of birds are migrating, not much happening. And then at the end of 2020, and since then, there have been huge numbers of viruses, virus confirmation in all sorts of birds. And over Europe wide, we've had over 2,000 outbreaks in poultry, which has meant over 46 million birds have been culled in agricultural, commercial establishments. And Europe-wide, nearly 3,000 events in wild birds. So it has been very, very significant. But the good news is, look at this right-hand corner of the chart. Back in June, in the summertime, the number of new positives went down very significantly. And we have now had the AIPZ zone lifted so that commercial birds can now go outside and farmers don't have to take as much care with their farm environments, which they've had. It's cost them a lot of money, even apart from the ones that were directly involved. The commercial aspect is where the money really is. And I remember 10 or more years ago, Hong Kong had an outbreak of a highly pathogenic virus, and they killed every chicken in Hong Kong. And you can imagine what that does to the local economy. So commercially, in establishments in Europe, and the more the less sophisticated poultry markets all around Asia are tremendously impacted by the occurrence of the highly pathogenic virus. It does kill the virus, the animals directly, but also all their contacts and are also culled. So huge impact. To clear up, a lot of biohazard material work is done. There is no risk to humans of catching the highly pathogenic virus from either eating poultry or from eggs, and that's proven over and over again. A very few number of humans have caught the virus, the avian flu virus, but they are people that have been very closely associated with either killing or cleaning up or indeed plucking infected birds. So there isn't a direct risk to human health from these birds. But what we are very, very aware of is the fact that the strategy that flu virus has for circulating is to mix various flu viruses and producing new strains. And occasionally a real change takes place. And I don't need to tell anybody here what a pandemic is, but flu pandemics we'll look at next are based on an avian flu strain mixing with a human flu strain and making a new pathogenic strain that passes human to human. And there was a real fear when the severe new avian flu outbreak occurred in 2003 that it could pass on. And I remember some of my last days in committee at work before I retired was writing out the pandemic protocols of what we would do to prepare ourselves for the next pandemic. And it is a completely natural virus cycle. The viruses circulate in all sorts of birds. They come in contact with each other. But of course, now domestic birds are in situations where they pass all sorts of infections very easily on to each other. They also come in contact with a particular species, swine, pigs, are very susceptible to flu, both from avian and from people. And of course, we come into this cycle as well. So it's a natural cycle, but because of the way the global population acts and the numbers of us, it is a more direct threat to our health than it's been in the past. But of course, yes, bird flu has gone away at the moment. It will be back. The birds are getting better, but now we have to start thinking about human flu. Man flu, seasonal flu, epidemic flu, pandemic flu. It seems to get us in all sorts of different directions. I'll get rid of man flu. Slight apologies for the popular name of the common cold. We know it very well. There's over 200 different strains of viruses that produce symptoms. Children are extremely good at passing cold viruses on. It's an upper respiratory tract infection. The virus uses the nasal mucosa and the throat to replicate and destroy and damage those cells, which give us the symptoms. And of course, there are four, as we know, 4 of the coronavirus strains are part of this common cold. This is not flu, so we move on. Oh. Seasonal flu. This is what is normal year on year, except for the last couple of years. We have new strains turn up and the virus drifts. It changes very slightly. But it is fairly similar year on year. There's also epidemic flu, where bigger changes take place from previous strains, and this is called, in virological circles, virus shift. And then there's pandemic flu, where we've got our completely new virus strain, associated with an animal usually, and the human population is susceptible. It doesn't seem long since in a few years ago, I had to explain in detail what a pandemic was. I can skip that this time. This is a WHO chart from Australia. The left-hand side is the summer of 2021, demonstrating that they had almost no flu. Well, it looks like no flu was recorded. They were cut off from the world, and they did not have a local outbreak. From January 2022, things started to change, more social mixing, a little bit of international travel, which has increased. And lo and behold, in the flu season, the normal winter of Australia, flu reappeared. And it's been quite significant in some places more than others. So the question is this going to be an indication of what is going to happen in the UK and Europe in our flu season? And as I've said, surveillance in the UK at the moment, there is almost no flu. So that's seasonal flu. if we go back to pandemic flu, we have very good records over the last century of what has happened. We know from clinically that there were pandemics in the end of the 1800s, but we don't know for certain which viruses were involved. Sorry about that. 1918 is the big first pandemic that we know a lot about virologically and also from the symptoms side. But there have been two other pandemics that are very well documented. In 1957, Asian flu, and it's quite possible that everybody in this audience was infected at that time. I seem to remember having the Asian flu. In 1968, Hong Kong flu, a completely different strain turned up. These were very different pandemics to the 1918 pandemic for reasons that I'll discuss. And 2009-10 has been labeled a pandemic, but I'll discuss that later. I'm not sure I agree with that label. We all know these pictures from 1918, a new virus in a world that was exhausted from a world war, that was still in the middle of huge population movements, troop ships coming and going. The numbers are just horrendous. No antibiotics, no sophisticated care of the sick, and the numbers are just huge. Even though the death rate doesn't look, statistically 3% doesn't sound much, but for a global population, it's really, really very significant. One thing I found interesting about this picture, in 1918, they knew the importance and the usefulness of wearing masks. And all of these nurses have masks on. The one person in the picture that does not have a mask on is the man who is who is probably driving the truck. I won't say anything else. And of course, huge numbers of people died, no antibiotics. It was a perfect storm as far as an outbreak could happen. The virus probably appeared in Western USA, possibly through pig contact. It spread from the States into Europe. It's called Spanish flu because it seems that the Spain, because it wasn't involved directly in the war, had newspapers that publicized it more than all the other European countries. So they were the first to publicize it in newspapers. So it got called Spanish flu. 1957, from this picture, it looked in the States, they had large numbers, but the numbers of people did not die. To think about the pandemic, the swine flu that turned up in 2009, 2010, we now know it has a very complex mixture of swapping genetic material initially from a human and avian mixture virus that then seemed to go through pigs and picked up a couple of two genes in the pig population, which was then passed to humans, which then became human to human transmission. It was first identified in Mexico, which was why it was called Mexican flu to start off with, and then became swine flu. And it spread around the world in a couple of days. Because it was an H1N1, some of the older population probably had some immunity, which is why I think it could be called an epidemic rather than a pandemic. But the important thing, apart from semantics, is that it had low pathogenicity in humans. And all the work that we had done planning for the next pandemic was in fact based on a new virus being highly pathogenic. But in this case, that did not happen. It did not cause severe illness. So again, flu surprised us. And I'm sure it will surprise us again in the future. Just a quick reminder about a virus. We have genetic material. This picture shows, a diagram shows the double helix of the DNA. Flu is an RNA virus, so it has a single strand of nucleic acid. All viruses have a protective coat, and they have structures on the outside of the virus particle to allow them attachment to their host cell. This delightful little chart has a meter-high human down here in the bottom left-hand corner. A human eye can see a hair, which is 10 to the minus 4. We then go to light microscope and we can see a cell, human mammalian cell, quite chunky. Bacteria, again, are 10 times smaller than the human cell. And then we get into the realm of viruses, 10 nanometers, 100 nanometers-ish, some are bigger, to 10, just over 10 nanometers. And they can only be seen by an electron microscope. This is a picture of an electron micrograph of influenza virus. It's what we call pleomorphic. It's got odd shapes, but what it is very well known for are the structures on the outside of each particle. This diagram shows what those particles look like or contain. The ribonuclear protein, genetic material in the inside, is RNA surrounded by a protein protective sheath. The virus, capsid, lipids, fats and proteins, and then the structures on the outside, which are absolutely essential to the life cycle of the replication cycle of the virus. In this diagram, hemagglutinin, which has 3 identical components, are stuck on the outside, and we know at least 9 different variations of this hemagglutinin. In this diagram, the pink is a tetramer, it's a neuraminidase, and we know of 9, again, at least, variations in the structure, subtle structure of the neuraminidase. And it is this mixing and matching of the types of neuraminidase and hemagglutinin which allows us to designate what a virus strain is. Influenza A is the most common and the most changeable virus. B and C are less of problems, and they don't change quite as much, they're more stable. This diagram shows nicely the hemagglutinin and the neuraminidase on the outside, but it shows importantly that the genetic material on the inside is in 8 different segments, and this again is underpins the strategy of influenza virus as being able to produce new strains that its hosts are not able to fight against, and it enables the viruses to mix and match with all sorts of other viruses. So the whole replication cycle strategy of influenza is replicate, but don't do it particularly efficiently so that new strains are produced and the host will not be won't be able to fight it. This is a nice diagram of the virus replication cycle in a cell. The attachment, which is through the hemagglutinin, it has to be a precise attachment receptor, and in this, for flu, it's sialic acid, which is a molecule that sits on all the cell membrane of all cells, not only mammals, but birds and insects. It's a lock and key. It has to match, the hemagglutinin has to match the sialic acid molecule precisely. It then gets engulfed into the cytoplasm of the cell. The virus particles get to the nucleus. They uncoat. The genetic material starts working and gives out the information for new viruses particles to be produced on the ribosomes of the cell. The ribosomes are the assembly lines of factories. So the cell is completely taken over to produce new virus particles. And once those new virus particle components are produced, they gather together to produce new particles. The new particle is, the release is facilitated by the neuraminidase, and thousands of these particles are produced per cell in 12 to 24 hours after replication. It's a completely passive attachment process. The more viruses that are introduced into the respiratory tract, the more likely it is to set up a successful replication cycle. And then this virus just gets passively expelled from the host, and hopefully, from the virus's point of view, passed on to a new host. The new host in our case is us. Children, again, are very good spreaders. I don't think my cough is a flu. I think it's a cold. The cough, the virus itself damages, destroys cells that line our respiratory tract, and the cough and redness and soreness is a direct result of that replication cycle. The other set of symptoms is caused by our immune system reacting to the virus infection, and that includes fever, tonsils swelling, lymph nodes swelling, the spleen getting activated, systemic chills and aches. This is all our immune system reacting to a foreign particle causing damage. Complications, of course, depends on how much damage the virus does. Children don't often get very ill with flu, but they can get ear infections, particularly, and sinusitis, and all of us are at risk of bronchitis. Less common complications are, of course, secondary bacterial infection, as well as viral pneumonia and brain and nervous tissue infection. Very rarely, the immune response does not come under control and sepsis occurs. Our immune system has two parts. There's an immediate, non-specific, innate infection response to an infection, and this is fever, and it's mediated by the white blood cells from the glands, and a very complicated system of non-specific but fast reaction to an infection. Then there's the acquired immune response, which again is mediated by different white blood cells, but also includes the lymphocytes that can produce antibodies. And this takes time. and it takes 10 to 14 days. And this is where vaccination comes in. We get a natural infection, we get natural antibodies, but if we get vaccinated against a particular infection, that primes our immune system. Sepsis, cytokine storm is especially occurs rarely in young adults, possibly because their immune system positive feedback controlling system isn't working quite so well. There are antiviral medicine targeted against specifically flu. They have to be taken very early on in the infection. Not very efficient, but they are out there and they are used for particularly vulnerable people, older people usually. Again, I have to always emphasize that antibiotics do not work against viruses directly, but of course they may clear up secondary bacterial infection should it occur. Vaccination. There's lots of pictures of happy, smiling people, all having had their flu vaccine. This is a promotional poster from 5 or so years ago. So who should have the vaccine? Well, in the past, children in clinical risk groups, but nowadays, for the last few years, all healthy children are given vaccine, and they were given it at school, as well as having to go to the GP for non-school age. Children we know are extremely good at passing on the flu virus. Very different to COVID, if I may just mention COV2 in passing. So we know that we can minimize transmission to other members of the population if we target healthy children to stop their transmission of the virus. Pregnant women, it's now recognized, do very much benefit from having the vaccine. It's perfectly safe in pregnant women, and it protects the baby for up to 3 months after birth. All adults that are at risk for their health issues are obviously should have the vaccine, and anyone over 65 years old is now eligible to have the vaccine. The strains are decided by the WHO for Europe back in February, March, looking at what's happened in Australia and Asia. It said that there were so few isolates available early on in 2022 that they didn't have as much information to go on to work out which strains to put in the vaccine. Vaccines are produced in lots of different ways now. The eggs, grown in eggs, is the traditional way. Cell culture is now used for producing live virus vaccine, and the new recombinant methods, molecular biology, are going to probably become more important, and they are on the horizon. We've probably all had texts and emails from GPs and from NHS and all sorts of ways telling us that we must go and get our vaccine. I think it starts being rolled out next week, but the sooner the better. It's available at walk-in clinics, at pharmacies, and at your GP. So this is one that we shouldn't be delaying our going to roll up our sleeves at. I put this rather quaint picture in just to comment on egg production of the traditional killed vaccine is on an industrial scale. The virus grows very nicely in the amniotic membrane and fluid of a fertile hen's egg, and this is where all the virus was grown for all the traditional viruses. And I did read somewhere where something like a quarter of the eggs in commercial egg production areas in the UK were for growing flu virus, not for us to eat. And of course, the most of the eggs that we eat aren't fertilized. Huge industrial scale. But children have a different vaccine, and they have a nasal spray. And it's a live virus that has been attenuated, has been adapted in cell culture. It only grows in the cooler temperatures that are found in the nose. The nose is at 33 degrees centigrade. And the virus has been passaged time and time again in cell culture so that it only grows in the nose. But you have to do that for each virus strain that you want to put in the vaccine. So it's quite expensive and quite, very expensive. Antibodies, special antibodies, IgA, as well as IgM and IgG antibody types are produced in the lining of the nasal mucosa, and it's those antibodies that that will react to a virus landing in the nose of a child. And it limits the virus replication to the nose. There are very few symptoms and no transmission from person to person. So magic puff up the nose, as we tell our grandchildren, make them feel good. And it can be given at school, but younger children from 2 onwards, it's worth having. And they get a certificate and probably a donut on the way home. As far as the traditional vaccine goes, it's only protein from the outer virus capsid. These are the virus strains that are in the current vaccine that we will be getting. There's a strain of H1N1, another up-to-date strain of H2N2. Note both of these are Australian strains, although they have been around a while. Flu B, although it doesn't cause very severe illness, it's an unpleasant illness in unpleasant symptoms in an adult. So these are the 4 strains that go into the adult inactivated, killed virus, cleaned up virus vaccine. For those of us over 65, it's the same strains. And for the first time, I think it is, we're having 2 strains of B, not just 1. But they add a naturally occurring oil, which is called an axis, an adjuvant. And it's basically a little irritant to our immune system. It is said our immune systems don't work quite as well as they used to, and the vaccine works better if you have this adjuvant, which rather stimulates the immune system and wakes it up to the presence of foreign particles that's just been injected into the muscle. So why vaccinate? Well, we prepare, we get the immune system ready to fight the virus infection immediately and specifically when we come into it. That's a good thing. It protects yourself. It protects your family. It helps the NHS. We're very used to that phrase now, aren't we? And it adds to herd immunity. And I've put herd immunity in inverted commas because it's only temporary herd immunity. Herd immunity means that everybody's immune, so the virus hasn't got many susceptibles. But of course, with flu, this is only true for that particular season. We have to keep doing it every flu season with a new strain so that we're able to be protected from the latest strain. which is very different from the traditional herd immunity with something like chickenpox or measles or rubella or polio, where the virus does not change, and what we have to do is just keep vaccinating the new members of our population, i.e. the children, because the rest of us have immunity. So yes, there is herd immunity for flu, but it's only temporary. We have to do it again the next year. So to end, is this what's going to happen to us? I don't know what the vaccination situation was in Australia. They have had a significant outbreak. It's been noticeable. Ours may be not as noticeable if enough people are vaccinated. Will we start wearing masks again on buses and in meetings if it starts to circulate? I'm thinking blinkered about flu. We know they work. We shall wait and see. We look forward to the season with interest, but also with a certain amount of care. And I certainly will be going for my flu vaccine as soon as, well, now that I have been offered it. So that's it, folks. I will stop sharing.
Wonderful.
Thanks. Oh, sorry. Yeah, thanks, Jane. That was fascinating. You covered so much ground, and it was particularly interesting to hear about the just how complex the way that sort of the transmission from one species to another can affect the emergence of new strains, and how we really have got to be on our toes all the time to pick these things up. What we're going to do now is to go to the comments and questions from the audience, and as usual, we're going to start with the comments in the chat box. So let me just get that up on the screen, and I'll go back to the beginning. First question I've come to, actually, you covered after the question was posted about Australia and how they get the strain, or the strains that they experience.
I mean, it is odd that the whole world thinks of Australia, and it must be said Southeast Asia, as the beginning of a flu season. Don't ask me why. Why don't we have the first strains? It's probably social contact and numbers of people, but yes, Australia and Asia are the beginnings of the flu season each year.
But it does just expanding on this point, it does beg the question that if the WHO has to make a call on what strains go in the Northern Hemisphere vaccine as early as February or March, which, if you think about it, is only autumn in the Southern Hemisphere, how, there's scope for error there. And I was wondering if some of the new technologies for vaccine production that we've learnt from COVID might enable that time lag to be reduced, so that we can wait a bit longer to get a better fix on the Southern Hemisphere ones before.
You'd have to ask the vaccine producers. I mean, the live virus, you can't. That is quite a long process to attenuate a new strain. So there are perhaps when it's all molecular biology and just a recipe of putting bits of molecules together in a mixture, it might be quicker, but at the moment that's not the case.
Okay, well, that's something for the researchers to get their thinking caps on. Moving down the list of questions, Ian in Weymouth had a question about something you mentioned during the talk about sepsis and the cytokine storm. He wondered what that was.
Well,
briefly.
The immune system reacts to any foreign particle, and it produces all sorts of chemicals. The white blood cells are activated. There's a whole range of different types of white blood cells. But as those chemicals and cells react with damaged tissue, there is a feedback loop, and eventually the production of the lymphocytes and the complement and all the different chemicals that are produced get shut down because the infection has lessened, damage is being repaired, so that everything closes down. But in sepsis and in a cytokine storm, that feedback loop doesn't work, and the immune system keeps attacking the cells of the host, and so it does more damage to the host than the virus did right in the beginning. It's quite rare. The cytokine storm is a label that was given to a lot of young people in the First World War that seemed to die not from the virus but from an uncontrolled immune response.
Okay, thank you.
Right. Ian, thanks you for that. Mike Hollingsworth asks, what structural changes are produced in the flu virus when it's attenuated?
It just means that it's not as good at multiplying within the cell. It probably, I'm not sure I know the structural change. It can't replicate it. It's temperature sensitive. It's not the virus itself. It changes its ability to grow at 37 degrees. That's the main change. And it will only grow in cells that are at 33 degrees centigrade. I'm not sure I know the molecular background to that, but that is the major change. The virus particle would be the same structure, but it's unable to cope with the higher temperature, so it is bound to be just in the nasal tissues.
Right. Okay. And he's got another question related, not related actually, but something that's obviously occurred to him. Dexamethasone, which turned out to be a real surprisingly effective drug in COVID, reducing inflammation. He's asking, any thoughts about whether dexamethasone could have the same effect on flu?
Well, it certainly could if someone was very ill and it was suspected that it was the immune system that was causing the problem, because it's a steroid that dampens down the immune response. So yes, I mean, it's used in all sorts of clinical situations where, I mean, we need the immune response at the beginning of an infection. And at the beginning of flu, let's say. We need our immune response to get working and to get the virus transmission within our epithelial cells and respiratory tract under control. But there comes a point where if the immune system is not being regulated properly, then possibly the steroids like dexamethasone would be of help. So it's a clinical decision somewhere along the line of infection and recovery.
Okay, thanks for that. Paula asks about vitamin D. Does it have any protective effect against flu, and if so, how?
Well, many things have. I mean, vitamin C, D. Being healthy is a good start to fighting any infection. There's no good evidence that any of those particularly have direct influence on virus replication and damage. It's a good idea to be as healthy as you can be. And if vitamin D and C are possibly lacking in your diet or your lifestyle, then by all means, top them up.
Sounds like good advice. Ken Carley asked a question that I think a lot of us have been wondering about over the last few months. He says he's booked his COVID jab for Saturday, and he's asking, does the COVID jab include the flu vaccine? I don't think it does it yet?
No, not yet, no. I'm sure a lot of pharmaceutical companies are working on that, but at the moment they're going to be two separate. As far as I know, they're definitely, all we are being offered is two separate vaccines.
Thank you.
Okay, that's Bernie and Kenneth is asking about the mass gatherings in London for the funeral and all the queuing up for the lying in state and everything. Are we likely to see a significant increase in flu because of this sort of thing?
Well, the baseline is very low at the moment, so there is hope that that will not happen. It is a bit early for flu to get going as well. Flu usually starts to increase in December.
Of course, masks are no longer compulsory or even advised, actually. So it'll be interesting to see how many, if any, people actually turn up to these things with masks. I know we're still wearing them. Here's Mike asked the same question actually about combining flu and COVID in the same. Oh no, it's not actually. He's asking a different question. I beg your pardon. I'll read it properly. He says, "Is it possible that flu and COVID viruses could combine to produce a new strain that humans haven't seen?" I wouldn't have thought so. Different, aren't they?
Very, very unlikely. I mean, they're two completely different organisms. They have different genetics. COVID is RNA, but it's a single-stranded one. No, I mean, it's possible that both viruses, it could be set up an infection in a person, but they are extremely unlikely. I don't think so, no. A person could be dual infected, certainly, but I don't think there's any real chance of the two viruses making a new hybrid between them.
Okay, yeah, that's a relief. Just one last, as it says, more as a comment actually. John Delafield says that, he says, for info, I had both the COVID vaccination booster and a flu jab a few days ago, one in each arm, with no adverse reaction.
Excellent to hear.
Yeah.
Very quick off the mark.
That's
so they can be, so presumably then it's been demonstrated that it's safe to give them together, but not necessarily within the same vial.
That is in fact what I'm expecting when I get my jab in early October, one in each arm, I think. Anyway,
it's possibly commercial as well.
Yeah,
you know, different people make different companies make different vaccines.
Yeah. Okay.
So thanks, Jane. That's the chat box dealt with. Thanks for everyone who put their questions in there. What we're going to do, I think, Steve, you're going to take over this bit.
Yeah, Robert, up there in Newcastle. Good morning, Robert. I know you've been waiting patiently with your hand up. Thank goodness it wasn't your real one. Go ahead with your question, Robert.
Yeah. Well, first of all, thanks, Jane, for a really erudite and interesting talk. Now, you did talk about the virus's strategy for spreading, which would sort of imply an intelligence, or is it just a way of talking about it?
Yeah. There's no intelligence. I apologize for doing that because I did not want to give that impression. No, I use the term strategy because that is the way just to describe how virus replication and transmission happens. No, that viruses have different. Evolution has produced different strategies for virus replication. Something like measles or herpes are very stable viruses. They're based on DNA. They have a completely different way of transmitting and keeping a new infection going. The strategy that has evolved for flu is rapid replication, lots of mistakes make, and it's got no proofreading in its replication of its genetic material, which means a lot of particles are produced that are dead ends. They don't go on. They're not successful in setting up a new infection. They're duds, if you like. But it also means that it has that new virus strains will be produced and will be available to infect new people over and over again.
All right, thank you.
Brilliant. Okay, thank you for that, Robert. Mike Trevithick from Thanet. Go ahead, please, Mike.
Hello, good morning. Yeah, thanks for your talk. I thoroughly enjoyed that. And just a comment about COVID and flu vaccines. We're here in East Kent. They're going to be separate injections at separate times. So I'm getting my flu vaccine this Saturday.
Yeah.
I've just had a message today to book my COVID vaccine sometime in the future. So you just want to check if you think you're going to get them both at the same time. Just check. Can I ask a question about the immune response?
I'll try to answer it.
What I'm most puzzled by, if you look, it takes antibodies a couple of weeks to generate antibodies to try and help us. Why is it so slow? What's the evolutionary advantage of having a response which is so slow? Because it means you're almost fighting if you're susceptible to get it. If antibodies could storm out in a day, presumably you'll be
any infected, I mean, it's not even infectious organisms that elicit a response. Pollen, any foreign particle can produce antibodies in the end. It's a question of a biological process. A flu virus infects the cells in the respiratory system. It damages those cells. It takes time for the white blood cells to recognize that there's an area of damage. The lymphocytes congregate around a damaged area. Information then gets, the white blood cells get stimulated to replicate. Then they send chemical messages to the lymph nodes in the neck, the spleen, under the arm, with identification of what this organism, this foreign particle looks like. And then the white blood cells have to be produced in large numbers. That are the organisms that then make the antibodies, which are a very small protein that are produced by lymphocytes. It's a biological process that takes 10 days. And obviously, evolution has never found a way of targeting material faster. Now, there's non-specific activity going on immediately. You know, you get a red swelling when you get a cut or something. It's immediate. So there's a lot of non-specific response, but to get a targeted response that matches that particular foreign particle, it just takes time.
Yeah. Okay. Thank you very much, Jane and Mike. Robin Clegg.
Many thanks, Jane, for a super talk, very informative. It sounded as if you worked before you retired in this area, and I just wondered what your job was exactly back then.
Oh, yes. Well, I was based at the Royal Berks Hospital in the clinical virology lab in pathology, but I actually was employed by the Public Health Laboratory Service, which was a national network of microbiology laboratories. So half my job was in the clinical lab at the Royal Berks in Reading. And, you know, every year we grew the virus. I mean, I was there quite a number of years, so early on it was quite slow going. We didn't have PCR and things to identify the virus. We would grow. Flu virus grows quite well in cell culture. It's a bit tricky to identify, but we would try and get throat swabs in from the hospitalized patients and also from GPs to grow the virus and then to identify it. And then we'd send it up to the head laboratory in Colindale of the Public Health Laboratory Service for them to type it. The Public Health Laboratory Service became the Health Protection Agency in about 2005, something like that. It then became Health Protection England, and some of it, the rump of it that is left, is now called the UK Health Security Agency.
Thank you.
Lovely.
I say no more.
Thank you, Robin. Right, nobody else has their hand up at the moment, and but in the chat box, our friend Jo Cork over there in Bristol, she just makes the point that vaccination against shingles is offered to older people, and I know this personally. I was offered it last year, and took up the offer, and fortunately, no after effects at all.
Yeah. It is such an important vaccine. Shingles can be really miserable, and anybody that has had chickenpox in their life, it still carries the varicella zoster virus in their nerves, their basal cell ganglia, and you're at risk of it replicating and causing shingles, and the vaccine is very efficient at stopping that. Yeah. So yes, thanks, Jo, for bringing it up. It's really important.
Yeah,
Can I just add something to that? I'm 80, and I should have been offered it when I was 70, but there was a period of about 4 months when the GPs had not caught up with the 70-year-olds. And then we were going to be offered it again when we were 79. And I only found out that because I had a double attack of shingles a few years ago, which was really quite bad. And they said, oh, you seem to have slipped through the net. So if you haven't been offered it and you're at my age, and you really you may want, if you can ask the GP about it, because you may just have fallen through the net and not been offered it. And then they're not going to offer it against once you pass 79. They're not thinking of offering it. So that's just a really.
Yeah. It's something that you can demand, and they should give it to you if you're over 70.
Good advice. Thank you. Robert, you're back again. On you go.
Yeah, sure. Joe, what's a double attack?
Well, I had bad shingles, and then I went to the doctor. Well, I went to the pharmacist, actually. It was great. I said, I think I've got some blisters on my back. And he took one look and said, go to the walk-in clinic. And I had it. And then a few weeks later, even though I'd had the vaccination, I had it again. And they said, this is quite unusual, but who knows what happens when you get old, and you shouldn't have it anymore. And I haven't had it again since, I'm pleased to say. And it was pre-COVID sort of thing. So, then I don't know. But that's all I meant was I had it twice, and people said, oh, you shouldn't have it twice. But they went, well, you have.
There's always an exception. I must say that there is also an antiviral that helps with shingles, but it has to be taken very early on.
Yeah,
it's called acyclovir.
Yeah. And when I went to the walk-in clinic, there was a young man there with shingles. Mine was on my back, and his was in his groin area, and he was in terrible pain. He was awful. It was very awful.
Yeah. You can get ocular shingles as well, and it's
And
really,
yeah. So mine was just superficial. It wasn't all that bad. But I remember hearing a lovely phrase once, which was a kind of joke, I suppose. What's the difference between love and herpes? Herpes lasts forever.
Go on, Barry.
Yeah, actually, Joe's remark about shingles and these cut-offs for whether you can get an injection or not prompts me to something you mentioned about the flu jab, about this business of over 65s get the jab that's got this adjuvant, this squalene adjuvant, and under 65s don't get it, but otherwise they're the same. Why doesn't everybody get the adjuvant if it makes the vaccine work better?
I don't know. I can only guess that one. A, finance. B, if you overstimulate when you give the vaccine, you might get local reactions more. If you give it to younger people, younger adults, I mean, there might be a more, energetic local response.
Yeah.
Those are the two things that come to my mind.
My GP a few years ago, my GP told me that the under 65s get a vaccine with 4 strains, and the over 65s, the most likely 3 strains plus the stimulant.
Yeah.
I don't know if that's right or not.
It was, but it's changed this year. We're getting 2 strains of B as well as everybody else. So it's only the adjuvant that is different.
All right. Okay. Well, I think that about wraps it up, Jane. Thank you so much for a really interesting talk. I've learned a lot. The graphics were terrific, and I really enjoyed looking at the sort of microscopic level, atomic scale almost. I've got something to say about that a little bit later, but first of all, please everybody thank Jane for a wonderful talk this morning. It's her second for us, so I hope she'll come back for more at some stage, but really fantastic. Thank you, Jane.
Thank you. Thank you. I'll wait for the next pandemic.